Up next, our next presenter is Megan Gliozzi from the U.S. Food and Drug Administration. Whenever you're ready, Megan.
Thank you. Hi, I'm Megan Gliozzi. I'm a postdoc in Dr. Heather Painter's lab at the FDA, where we study transcriptional dynamics throughout the blood stages of Plasmodium falciparum's life cycle. In this particular project, we aimed to identify the RNA-protein interactome throughout the asexual stages of development. Next slide. And we chose to study the RNA-protein interactome in this study specifically because research has strongly suggested that regulation of stage-specific gene expression occurs primarily at a post-transcriptional level, causing a just-in-time type of gene expression and really emphasizing a critical role for RBPs or RNA binding proteins to both regulate and stabilize RNA until the exact moment gene expression is needed. Unfortunately, due to a lack of effective in vivo methodologies, both this type of regulation and RNA-RBP interactions in general are not very well characterized in Plasmodium. So we set out to expand our knowledge by capturing the interactome every 12 hours of the 48-hour IDC so that we can identify RNA-RBP interactions at the ring, early and late trophozoite, and schizont stages of parasite development. Next slide. In order to identify the interactome in this study, we're utilizing the PARCL method, which is an advanced cross-linking method that increases both sensitivity and specificity of RNA-protein interaction capture compared to conventional cross-linking methods. This method also, in addition to identifying RBPs, can allow us to identify RNA binding site footprints within transcripts because each site of cross-linking actually generates a T2C mutational signature within the RNA that can be assessed in downstream analysis. So starting from left to right in our schematic, we start our experiments at each time point by incubating our parasites with a modified nucleotide, 4-thioluracil or 4-TU, to allow its integration into nascently transcribed RNAs. This is a really critical step for the PARCL method because the thiol group of those modified nucleotides integrated into our transcripts is what actually cross-links to zero-distant proteins at the PARCL specific 365 nanometer wavelength. After we've cross-linked, we can lyse our parasites and run that lysate over a silica column to isolate all species of RNA and any cross-linked proteins. After several washes, we've isolated our RNA-RBP complexes and can next either treat with RNases to digest any RNA away and focus on RBP identification by mass spectrometry, or we can treat with proteases to remove protein and look for RNA binding footprints through mutational analysis using RNA-seq. Next slide. I'd like to start. Oh, sorry, that's fine. We're going to start by looking at RBP identification in our time course. Next slide. Across all 4 of our time points, we identified a total of 423 RBP candidates that were significantly enriched in our PARCL sample compared to our control. As you can see on the graph on the left, the number of RBP candidates identified at each stage varied quite a bit, with the most unique and most overall RBPs identified in the late trophozoite stage. The Venn diagram on the right, we compared our RBP candidates to what has been previously annotated as RNA binding. And we found a substantial proportion, 210 proteins, that have been previously reported to bind RNA, leaving an equally substantial proportion, 213 proteins, that have yet to be annotated for any type of RNA interaction, which really made us start to wonder what types of proteins and what types of protein functions are we really identifying in this candidate list. Next slide. So to get a better idea of the protein functions that we find in our RBP candidate list, we next performed gene ontology or GO term enrichment analysis by stage. And we do find a lot of GO terms enriched in our data set that suggest RNA binding, which makes sense for a list of RNA binding protein candidates. These types of terms include several types of RNA binding, RNA processing, association with ribosomes, and translation. Next slide. But we do also find GO terms enriched in our data set that don't necessarily suggest any direct interaction with RNA by name alone, such as DNA binding and chromatin organization, nuclear transport and protein dimerization, as well as several metabolic and enzymatic processes, really suggesting that in addition to canonical type RNA binding proteins, our method is also helping us identify non-canonical type RBPs that could either be regulated by RNAs or moonlighting their primary function to regulate RNA on the side. Next slide. Switching gears, I'd like to next talk about our pilot study analyzing RNA binding footprints in the late trophozoite stage. Just a little reminder, since it's been a while since the methods slide, this type of analysis is looking at generated T to C mutations with RNA sequencing. Next slide. And before we really started to look at the types of RNA footprints we could identify, we first wanted to see if these types of these T to C mutations were truly experimentally induced. And looking at the two graphs on the left, we found our partial sample in blue when compared to our control in black had a higher, pretty significantly higher overall T to C mutation rate. And at each T to C mutation site had a significant average mutant allele frequency, much higher than our control as well. Both of which really suggest that the mutations we're seeing in the partial sample are likely experimentally cross-linking induced rather than any type of sequencing error or strain-specific SNP. Looking at the graph on the right, where we're looking at the number of T to C mutations within a gene body for genes that had at least one T to C mutation in our experiment, we find our partial sample, again, has more T to C mutations per gene body compared to our control. You can see our control in black has 1, maybe 2 T to C mutations per gene, whereas the partial sample has T to C mutations ranging all the way up to 20 mutations within a gene body. And when you really think about RNA binding proteins interacting with a given RNA at any particular point in time, you'd expect that they'd be interacting with more than just one nucleotide at a time, which should correlate to more than one T to C mutation within a transcript or gene body. So the fact that we're seeing a lot more T to C mutations per gene in our partial sample compared to our control further supports that the mutations we're seeing here are experimentally induced and likely indicative of RNA-RBP interaction. Next slide. Pretty confident in our T to C mutations. We next wanted to get an idea of the patterning of these mutations to see if we could identify any potential RBP functional interaction types based off of where the T to C mutations lie within the transcript. And we found a lot of transcripts that had T to C mutations similar to this top gene expression track, where they're very localized within a particular spot of a transcript body. To us, this kind of suggests maybe a single RNA binding protein interacting with that transcript in a stabilizing manner. And when we actually look at this particular example, expression profile in a previous publication by Heather, where she looked at transcription versus stabilization at every hour of the IDC, we can see at our late trophozoite stage of 36 hours post-invasion that indeed this particular gene is more stabilized than it is transcribed. Alternatively, we do see different transcripts that have a pattern more similar to the lower gene track, where we see the T to C mutations are much more dispersed across the whole gene transcript, sometimes even at the point of a splice junction. To us, this suggests maybe more than one RNA binding protein interacting with this particular transcript, maybe in the process of transcription or post-transcriptional modifications, suggesting genes with this type of pattern are more so transcribed than they are stabilized at a time point. And indeed, for this particular gene, we're showing, again, looking at the expression profile, this time we see a more transcription type profile. All in all, suggesting that we can get a sense of the type of RNA-RBP interactions occurring based off of where T to C mutations lie within a transcript. Next slide. So in conclusion, we have expanded the characterization of the RNA-protein interactome throughout all stages of the IDC. And we've provided the field with a new method for in vivo RNA interactome capture and identification. Our proteomic analysis has identified both canonical and non-canonical type RBPs, demonstrating this method's ability to identify all classes of RBPs. And our RNA sequencing analysis has identified what we believe to be functional mutations of cross-linking RBP RNA interactions. And while we don't believe a global study like this will provide a consensus binding sequence for any of these types of interactions, we do think that scaling down to a single RNA or RBP of interest very well could create or generate a consensus binding site in those cases. Our ongoing studies include further data analysis of our mass spectrometry data to not only identify the RBPs, but the RNA binding regions within those RBPs, follow-up studies with RBP candidates of interest, and next slide. Expansion of the RNA interactome into the sexual gametocyte stages of development. Thank you to my lab, our collaborators, and all of you for your attention. I'll take any questions.
Dr.
Very impressive hardware and software. You're going to get a lot of candidates. How do you anticipate identifying the one that's interesting?
Hi, I'm turning my volume up. I'm sorry, I had trouble hearing that. Could you repeat yourself?
Yeah, you have very interesting candidates. How you anticipate which ones are going to be like the most important ones?
So we're picking things based off of either the fact that they're non-canonical or historically they may have some suggestion to essentiality. But I think really in order to identify how critical any of these are, we'll have to do follow-up studies with that RBP, tagging it, pulling down, seeing what RNAs are actually interacting with it, also doing knockout type studies. And we do have a good, pretty long list of RBPs of interest that we're working on right now, but until we actually get the data, it's hard to tell. Great question, though.
You're gonna have to be lucky, but luck happens.
Yeah, that's why we keep going, right?
Right, go for it.
Thank you.
Do you have any additional questions? No? Okay, good. Thank you, Megan.
Thanks.